Okay, I'm seeing lots of participants coming into the webinar. I just want to say hello and welcome, and we're going to wait a few more minutes to get started. If you want to let us know in the chat what cytokine or disease, inflammatory disease that you're studying, that would be a really fun way to engage with us. If not, just sit back and we'll get started in a few minutes.
Clark, nice haircut.
Thank you.
Sure.
All right, just a couple more minutes as we have more attendees filtering in. Welcome, everyone. Thanks for being with us today. So we have some more people joining. Perfect. Yeah, welcome everyone. So it's 1 o'clock. I'll just go ahead and again, welcome everyone to today's webinar. Good afternoon. We're very excited to talk to you today about our inflammatory disease models that can hopefully help you discover and test novel therapeutics targeting cytokines and receptors. So before I jump in, I'll quickly introduce myself and our panelists. So I'm Jenna Frame. I'll be moderating today's webinar. I have a PhD in pathology and roughly 10 years of experience working with various genetically modified mouse models. And currently I manage our scientific communications and marketing here at Biocytogen. I'm joined today by our speaker, Dr. Clark Gasper. Clark is a senior business development manager here at Biocytogen Boston. Clark's 13 years of research experience covers a broad range of fields, including protein biochemistry, antibody production, and small molecule drug development. Clark earned his PhD in biochemistry from Boston University, where he worked on identifying novel lead candidates for the treatment of diabetes and obesity. And also joining us for today's Q&A session is our CEO, Dr. Ching-Hong Lin. Dr. Lin has extensive research experience in molecular genetics and has held numerous leadership positions in the industry. He served as Senior VP at Biocytogen as well since 2018. So welcome to our panelists and our speaker. And without further delay, I would like to invite Clark to begin the presentation. And I just want to quickly say that we will take questions and make some announcements after the presentation. So please stay tuned. Take it away, Clark.
Thank you, Jenna, for the introduction. Good afternoon, everyone, and thank you again for joining us for our December webinar. I'll start by providing a brief introduction to our company and the services we provide. Biocytogen was founded in 2008 in Worcester, Massachusetts. We initially focused on genetic engineering, where we developed proprietary methods that improve gene editing efficiency, even for very large insertions. And so with this platform, we've been able to supply over 3,000 custom animal models for both academic and industrial clients. We have since expanded our service platforms to include preclinical pharmacology and therapeutic antibody discovery, which uses our humanized immunoglobulin mouse platforms, RenMab, RenLite, and RenMab Hits. And so we use these integrated platforms of gene editing, animal models, antibody discovery, and pharmacology to help accelerate drug discovery for our clients worldwide. As an outline for today's presentation, I'll start by introducing the immune system, cytokines, and their role in autoimmune diseases. I'll then go into modeling inflammatory diseases in mice, including asthma, atopic dermatitis, and psoriasis, each with specific case studies. And finally, I'll summarize how these models can be integrated into drug discovery platforms and can help streamline your therapeutic drug discovery workflow. So in the immune system, responses to antigens, both self and foreign, are tightly controlled in a healthy individual, dictating survival, maintenance, and growth of immune cells. After a foreign antigen is recognized and removed, these cells normally stop dividing and become memory cells or are cleared from the body. However, a lack of proper inhibitory mechanisms can lead to allergic responses and inflammatory diseases. Similarly, a lack of inhibitory mechanisms for these immune cells can cause overproduction of lymphocytes and abnormal responses to self-antigens, resulting in autoimmune diseases. And so autoimmune diseases have a complex pathophysiology and are thought to arise from a combination of genetic or epigenetic factors and various environmental triggers, such as infection, smoking, and diet. These initial triggers can cause abnormal or defective immune responses that can cause a decrease in the expression of self-antigens by the thymus. And so this leads to a reduction in the activation threshold for self-reactive lymphocytes, is resulting in their elimination or inactivation. And so over time, these self-reactive lymphocytes persist and contribute to high levels of autoantibodies or cytokine imbalances that eventually lead to disease. In addition to autoimmune diseases, there are a few inflammatory diseases associated with Th2 immune responses, which is a pathway that focuses on maintaining barrier immunity. These pathways, however, can become overactive in response to various allergens. One example is with atopic dermatitis, where immune activation causes cell infiltration into the skin. So this results in bright red skin that is thickened and scaled, causing impaired skin barrier function. So in the case of asthma, an overactive Th2 response in the airway results in excess mucus production and a contraction of the smooth muscle, resulting in airway obstruction. So interestingly, these diseases, along with other Th2 diseases, such as allergic rhinitis, food allergies, and conjunctivitis, are strongly associated with one another. Therefore, targeting the common inflammatory mediators of the Th2 disease is of significant clinical benefit. So other factor that plays an important role in cell signaling are cytokines, which are small proteins associated with the immune system. There are several different autoimmune and allergic diseases that have been associated with specific cytokines. So as you can see, many of the cytokines overlap between diseases, and these cytokines have been targets of interest for discovery of new therapeutics. So in order to test the potential of these therapeutics in a preclinical setting, Biocytogen offers disease models and humanized cytokine mice to help validate drug candidates faster. And so this brings us to our first poll question, which is which cytokines are associated with Th2 immune responses? And so this question should pop up on your screen in just a second. And so we'll give about 20 seconds to answer. Great, so we'll close the poll. And the answer is D. The Th2 type cytokines include interleukin 4, 5, and 13. And so these cytokines are associated with the promotion of IgE antibodies, as well as proliferation of eosinophils. And so in excess, Th2 responses will counteract the Th1-mediated antimicrobial actions. So the optimal scenario is a well-balanced response between Th1 and Th2. So for the second part of the presentation, we'll be focusing on modeling 3 diseases in mice: asthma, atopic dermatitis, and psoriasis. For each of these models, we'll provide a proof of concept study introducing the disease model, followed by efficacy studies performed in both wild type and humanized mice. And so asthma is a common lung disease affecting more than 3 million people in the US alone. It's likely due to a combination of environmental and genetic factors and is triggered by various substances. Symptoms include difficulty breathing, chest pain, cough, and wheezing due to constriction of the smooth muscle in the airway as a result, as well as the big buildup of mucus. So at Biocytogen, we model asthma in mice using ovalbumin, which is a protein found in egg whites. This acute asthma model has been validated in different strains of mice to test for the efficacy of novel asthma therapeutics. And so in this model, mice are first sensitized by IP injection of ovalbumin and the adjuvant aluminum hydroxide. This is then followed by a challenge phase where the mice are nebulized with ovalbumin for several consecutive days. Treatment of the mice with therapeutics can begin on days 20 and 23, as shown in the dark blue box. And so to confirm the induction of asthma-like phenotypes, immune cells were quantified in the bronchioalveolar lavage fluid, abbreviated BALF. These assessments reveal a significant increase in the numbers of CD45 positive cells and eosinophils in the asthma-induced mice, which is shown in red compared to the control group in black. In addition, the ovalbumin-treated mice showed a significant increase in serum IgE levels and have asthma-related pathological changes, as demonstrated by immune cell infiltration and increased levels of mucus. And so these results indicate that ovalbumin successfully induced asthma in these mice. In this second study, asthma was induced in BALB-C mice using ovalbumin, and dexamethasone, which is a steroid that reduces inflammation, was also given at various doses. And so, as in the previous study, ovalbumin significantly increased the proportion of eosinophils as shown in group 2 with the red bars. However, these numbers were dramatically decreased in the animals that were treated with dexamethasone, as seen in the red boxes. And so higher doses of dexamethasone also reduced serum IgE levels in asthmatic mice compared with ovalbumin alone. And on the right, H&E staining demonstrated that the inflammation and mucus significantly increased in the ovalbumin group only. And so this demonstrated successful modeling of the disease. And this study also showed that in higher concentrations of dexamethasone, there was a reduction in inflammatory infiltration and mucus secretion, demonstrating that this model can be used to test the efficacy of this corticoid steroid treatment. And so in order to test the efficacy of novel IL-4 targeting drugs for asthma and other inflammatory diseases, Biocytogen has generated and validated a double humanized IL-4 receptor model. And so on the left, levels of mouse and human IL-4 were measured in the serum of black 6 mice and the humanized IL-4 mice. The black 6 mice only express mouse IL-4, while the homozygous humanized mice only express a human IL-4. We also ran a flow cytometry analysis, and stimulated T cells isolated from the humanized mice only express human IL-4 receptors on their surface. In this third study, asthma was induced in our double humanized IL-4 mice, which also exhibit similar changes in the number of immune cells and eosinophils in the BALF. However, in this study, the addition of Dupilumab in the green group 3, which is a monoclonal antibody therapy that blocks human IL-4 receptor binding, was administered and showed that immune cell numbers and eosinophils were significantly lowered compared with the disease-only group in red. And so lastly, on the left, Dupilumab treatment reduced serum IgE levels, and with the H&E staining on the right, it indicated that pulmonary airway had reduced levels of mucus production and infiltration of immune cells. And so these results demonstrate that ovalbumin can successfully establish the asthma model in our double humanized IL-4 mice, and that this model can be used to evaluate the efficacy of novel IL-4 targeting drugs. And so this brings us to our second poll question, which should pop up shortly. Which common allergen is associated with asthma? Go ahead and end the poll. And so the answer is E, all of the above. And so pet dander is commonly associated with skin cells that are shed from our furry friends. Pollen is the powdery substance shed by plants, usually by trees in the summer, grasses in the spring, and weeds in the fall. And then mold releases spores that can be inhaled, while dust mites are microscopic pests that commonly live in house dust. And so all of these contain foreign proteins that can aggravate asthma symptoms. And so another disease we have modeled in mice is atopic dermatitis, or AD. So atopic dermatitis is a chronic skin disease that presents with itching, redness, dry, cracked skin, and small raised bumps that can become infected if irritated. They can appear anywhere on the skin, and repeated scratching can cause thickening and hardening of the skin. AD is thought to stem from a dysregulation of the immune system, causing the production of inflammation that damages the skin barrier. And so there's no cure for AD, and treatments consist of topical steroids and behavior modifications. At Biocytogen, we've developed a protocol to model atopic dermatitis in mice, which has been validated again in several strains. In this model, skin lesions are chemically induced using oxazolone, beginning with a sensitization period on day 0. This is then followed by challenges 3 to 4 times a week from day 7 to day 26. And treatment of the mice with therapeutics can be given twice a week, beginning on day 6. And so in this model of AD, ear thickness of the mice is monitored, as it should increase in response to oxazolone, and serum IgE production is also measured. Looking at the data in red, both show a significant increase compared to the vehicle control in black. In addition, histology analysis on the right demonstrates that oxazolone induces stromal cell proliferation and inflammatory cell infiltration in both the dermis and subcutaneous tissue. And so this is confirmed by quantification of eosinophil infiltration here. In this second study, the efficacy of dexamethasone was assessed in the AD model. And so ear thickness and serum IgE levels were measured again, and different concentrations of dexamethasone significantly reduced ear thickness compared with the oxazolone-only group. However, these high doses of treatment, as shown by the dark blue line, also led to significant weight loss compared with the control group. And then thirdly, serum IgE concentration suggests that higher doses of dexamethasone may reduce the overall concentration of serum IgE levels compared with oxazolone alone. Additionally, H&E staining of the mouse ear tissue sections again indicated pathological changes related to atopic dermatitis, including thickening of the dermis and mixed inflammatory cell infiltration. However, these phenotypes were significantly alleviated in the dexamethasone treated group, especially in the high dose group, which returned to baseline. The eosinophil infiltration score in the skin was also significantly lowered in the dexamethasone treated groups. And overall, dexamethasone improved atopic dermatitis related symptoms, with the high concentrations having the most efficacy. And so in this final AD study, the efficacy of the anti-IL-4 receptor antibody Dupilumab was assessed in an AD model using Biocytogen's double humanized IL-4 receptor alpha mice. And so measurements of ear thickness on the top right indicate the highest concentrations of Dupilumab, shown in light green, could improve ear skin edema compared with the oxazolone and isotype control group, as shown in red. Similarly, high concentrations of Dupilumab significantly reduced serum IgE concentrations, as shown in the red box. And so these results suggest efficacy in mitigating AD phenotypes. Lastly, Dupilumab treatment alleviated some of the pathological changes related to atopic dermatitis, specifically eosinophil infiltration score. And so together, these results indicate that our double humanized mice provide a powerful preclinical model for in vivo efficacy evaluation of anti-human IL-4 receptor alpha antibodies. And so the third disease model I'll be presenting is psoriasis, which is also an inflammatory skin disease. Similar to atopic dermatitis, psoriasis is a chronic skin disease that causes red itchy patches with scales presenting, most commonly on the knees, elbows, trunk, and scalp. Symptoms tend to go through cycles, flaring for a few weeks or months before subsiding. Treatments are available to help manage symptoms, but there's no cure. Psoriasis is caused by an overactive immune system, which speeds up normal skin cell growth. And so typically skin cells grow, shed, and turn over in a 30-day period. However, with psoriasis, the overactive immune system causes skin cells to do this in only 3 to 4 days. And so instead of shedding, the skin cells pile up on the surface of the skin. And so here is a simplified mechanism of the psoriasis pathway, highlighting the 3 main cytokines associated with psoriasis, IL-22, 17, and 23. At Biocytogen, we've developed a psoriasis model in mice to test for efficacy of novel therapeutics. And so in this model, skin lesions are chemically induced with Imiquimod, which stimulates the overactive immune system in the area where it's applied. Imiquimod is applied topically and once daily for 5 to 8 days on the dorsal back skin. Treatment of the mice with therapeutics can then be given 3 times on day 0, 3, and 6. And so in this initial model build, Imiquimod induced a psoriasis-like phenotype on the dorsal skin after 4 days of exposure. The erythema and scaling of the skin were scored individually and then cumulatively. And they indicate that Imiquimod induced skin inflammation that was at its peak after 4 days and then slightly decreased. And so in contrast, the shaved mice that were treated with a control did not exhibit the phenotype. Histology analysis indicates that Imiquimod induces keratinocyte proliferation and infiltration of inflammatory cells. And so these results increased epidermal thickness and skin lesions. And so taken together, these results indicate Imiquimod can induce a psoriasis phenotype in these mice. Next, we examined the potential efficacy, again, of dexamethasone on improving the psoriasis phenotypes. Psoriasis was induced with imiquimod cream, causing erythema and scaling in the red untreated group. And so these symptoms were alleviated by treating the mice with higher doses of dexamethasone, as shown by the orange line. Histology analysis of these mice indicates that dexamethasone inhibited keratinocyte proliferation and inflammatory cell infiltration in the skin, similar to the control group. Epidermal thickness and overall histopathology scores also indicate that the dexamethasone prevented psoriasis-like injury and skin inflammation in a dose-dependent manner. And so in order to test the efficacy of novel anti-human IL-17 drugs, Biocytogen has generated a double humanized mouse that expresses human IL-17A and IL-17F cytokines. And so to confirm the secretion of IL-17A, a serum was collected from mice that were stimulated with anti-mouse CD3 and anti-mouse CD28 antibodies in vivo. The serum was then analyzed via ELISA with a species-specific IL-17A ELISA kit. And so on the left, mouse IL-17A was only detectable in wild-type mice, while human IL-17A was exclusively detected in the homozygous double humanized IL-17 mice. Similarly, to validate the expression and confirm the secretion of IL-17F, CD4 positive T cells were sorted from splenocytes and induced into Th17 cells using PMA and ionomycin. And so the results on the right show that mouse IL-17F was only detected in the supernates from wild-type mice, while the human IL-17F was exclusively detected in the humanized mice. And so to test the efficacy of novel antibodies targeting the human IL-17 pathway, psoriasis was induced with imiquimod over a 6-day period. Beginning on day 0, Bimekizumab, an anti-human IL-17 antibody, was intermittently dosed at both low and high concentrations. And so shown in the green and blue curves here, the anti-IL-17 treated mice have an overall reduction in skin erythema and scaling. In addition, histology analysis showed that higher doses of the anti-IL-17 treatment reduced disease severity via epidermal skin thickness and histology scores. And so together, these results show that Bimekizumab prevented psoriasis-like lesions in our double humanized IL-17 mice in a dose-dependent manner. And so this brings us to our final poll question. Which monoclonal antibody therapy targeting TNF-alpha was first approved by the FDA for treatment of moderate to severe psoriasis? So this question should launch now. Okay, we'll go ahead and end the poll. Share the results. And so the answer is A, Infliximab. Infliximab is a mouse-human chimeric monoclonal antibody that binds to TNF-alpha. And so this prevents TNF-alpha from interacting with its receptors on the cell and can mitigate these autoimmune reactions. It was first approved by the FDA in 1998 for IBD and Crohn's disease. and then later approved for treatment of psoriasis in 2006. Okay. And so lastly, I will briefly introduce other humanized models that can be used for disease modeling. So in addition to the humanized models used in the studies shown today, here are some other humanized models that may be of interest for a single or dual targeting of cytokines and chemokines that are associated with psoriasis. So in addition to the first two models, which we outlined today, we have TNF alpha with IL-17A, IL-23A with IL-12B, and CCR6. And so if you have a broader interest in immuno-oncology targets, there are many other strains of humanized immune checkpoint mice that we offer. And so we welcome you to visit our website for the full list of strains, including multigenic humanized immune checkpoint models. You can also download our full catalog using the QR code at the bottom of the screen, or you can reach out to us after the webinar and we'll be happy to assist. So lastly, I wanted to mention that we have started building a tissue library that as a companion to our humanized mouse catalog. The library consists of tissues from our various humanized mice as a way of providing samples to researchers for validation assays before having to commit to either a large animal order or a range shipment of live mice. So we currently have tissues available from these 5 models and are accepting requests for other models from our catalog as the library continues to grow. And so if you're interested in a tissue sample from our library, feel free to reach out to us, and again, we'll be happy to help. And so with that, I would like to thank you for your attention, and I'm happy to answer any questions in the following Q&A session.
Thank you so much, Clark. Let's see. Looks like we have a few questions rolling in. So in the first question, in the AD study with dexamethasone, do you know why the high dose leads to a decrease in body weight?
Yeah, so I can take that question. It is typically thought that dexamethasone induces oxidative stress in cells by increasing reactive oxygen species. But beyond that, the mechanism is not really known. So more research into that oxidative stress pathway would be needed.
Okay. You think it's independent of food and water intake?
Yeah.
All right, so next question is related to the psoriasis model. There's a question whether we see elevated IL-12P70 in the IMQ-induced psoriasis. Clark, do you happen to know, or Dr. Lin, whether
Yeah.
IL-12?
He may mention about IL-12, P40.
P70.
Right, IL-12 have P40 and P35. So P70 sometimes we come as a complex.
Okay.
Clark.
Yeah, so we haven't specifically measured those readouts, but that's something that we're open to doing for these mice for further validation. Right,
and we do have humanized IL-12 models as well, so that would be relevant to explore.
And for IL-12, we have double for cytokine and double for receptor, and also quadruple for cytokine and receptor. So welcome to use this model for your study.
Yeah, absolutely. It's important with these cytokine models that, you know, you sometimes have to humanize both the cytokine and the receptor because of the complexity of the signaling pathway. And so, you know, we've tried to do that for you so you can, you know, get the model and just start your study right away. So next question. We talked about specific reagents such as ovalbumin and oxazolone for inducing these diseases. Have we explored any other methods for inducing these diseases?
Yeah, so some of the other models that we are working on building are asthma using house dust mite extract and a colitis using dextran sulfate sodium. In addition, we regularly partner with our clients to develop new disease models, beginning with an initial model build to gather initial readouts and help frame a protocol. And so if you're interested in building a specific disease model, just let us know and we'll be happy to explore building it with you.
Yeah, that's great. I think our, I was going to mention it after, but our next webinar, which will be in January of next year, will cover some of these newer disease models. So please stay tuned for more information about that. Okay, moving on to the next question. Have you experienced any limitations with these models?
Yeah, so in terms of the skin diseases, we've noticed that a large area is required to induce serum IgE levels. But other than that, not so much with the skin models. However, with asthma, one challenge is with the BALF collection. And if you're also interested in lung histology. And so collection of BALF can sometimes damage the lung tissue in the process of collection. So if histology of the lungs is a preferred readout, we would suggest omitting BALF to preserve the lungs for histology.
So next question is, how do you determine the frequency of dosing with your test articles?
Yeah, so usually our clients will have initial data that helps inform a dosing frequency of their test article. But if they're unsure, then we usually do a literature search to find previous dosing schemes, or we can suggest starting with a dose response study or maximum tolerable dose before proceeding to efficacy.
And Gina, over here, usually the client come to us with a lot of in vitro data, and that data set help us to determine their dosing frequency and also dose.
Okay, yeah. So the next question is, how do the humanized mice behave in response to various stimuli as opposed to wild type mice? So do their respective immune responses line up where the humanized gene is involved?
Yeah, so that's one metric that we try to evaluate when we validate these mice. And normally we don't see any differences, both in terms of stimulation as well as immune cell profiles in the wild type versus humanized mice.
I agree with Clark. When we promote the model, that means we have been doing the phenotypic analysis, including the RNA expression, protein expression, and immune system, particularly when we're talking about the cytokine receptor, because they have been shared so many units, like either cytokine subunits and receptor subunits. So humanization is very tricky without interrupting other cytokine pathways. So we do solo study. But again, we need to pay particular attention when you do your study, because other cytokine pathway may get influenced a little bit.
Right,
Yeah.
and also that's why we try to, keep the internal part of the signaling pathway intact, right? So we're targeting the extracellular part so we can receive the signal for the humanized cytokine, but then the intercellular part might be the murine part of the receptor.
That's one way, Gina. And another way, the problem is because they share subunits.
Right.
Mm-hmm. So receptor subunit beta or gamma sometimes have been shared by multiple cytokine pathway. Therefore, we humanize four row, but then these subunit will be shared by other cytokine receptor that can influence a little bit. But luckily, so far, we didn't see that phenotype strongly because highly conserved between mouse and human, luckily.
Right, yeah, that's a good point. Let's see, okay. So our next question, do we have mouse models for other immune diseases like MS? And are we available to work with firms looking to work on neuroimmune diseases and neuroinflammation?
Yeah, so as far as I know, we don't currently have an MS mouse model, but like I mentioned, we're constantly working with clients to develop new models. Some of them, they are exclusive to our clients, so we don't have that information to share, but we use our expertise in order to work with our clients to build the model of their choice.
For brain tumor, actually, the challenging is the skill, is the surgery skill or injection inoculation skill. And luckily, our scientists, the Langao, have very good experience of their area. So most of these can be applied to client needs. But in general, you should talk to Clark and our team before you initiate that study. And our team definitely can try our best to help. And I saw we have been got request and support multiple brain tumor study because of the unique skills. But usually that cannot be scaled up quickly. So we highly recommend to using our humanized mouse model because that one, the cohort can be larger, study can be more reproducible. So humanized mouse model is very useful for your study.
Yeah, I would like to mention, as someone mentioned in the chat, that we do have an EAE model that's well established, but yeah, I mean, as far as other specific models, yeah, we will work with you. Okay, so next question. IMQ induces both Th2 and Th17. Did you test whether Dupilumab works in your humanized model?
So far, we haven't done the test in the IL-17 mouse yet.
Okay. Another general question about humanized mice. Do we ever see spontaneous death or any health problems? No, that we should note.
Yeah, no.
Let's see, another specific question. Any plans for eosinophil mediated esophagitis model? We might have to get back to you on that.
We don't at the moment. We don't.
Not at the moment. Okay.
Yeah.
Yeah. And we have another question about looking for more details about the IBD model using DSS. Is that using the human cytokine background?
Yeah, so we're starting to explore this model in both wild-type mice and our humanized, I believe it's the IL-4 mouse. And this usually takes the form of administering the DSS in the drinking water at about 3, sometimes 4% over a series of 7 days to acutely model the disease. And so some of the readouts for that model are like the colon length, as well as blood in the stool.
Clark, here actually we have 2 types of concentration for the chronic or acute disease. So depends on the concentration we use, 2% versus 3%, 3.5 or 4% even, that's high. So when we go down to 2%, then the window is much larger and the disease is not aggressive. Depends on client need, we do have both.
Okay, that's great, thank you. See, any more questions? Let me just check the chat. Okay, looks like we've addressed all the questions for now. But I would, like to remind everyone that you can reach out to us at any time using the contact information here. You can email us at info@biocytogen.com. Check out our website, but if you can't find what you're looking for, just feel free to reach out to us and someone will be in touch with you. And so before we end, I just want to make a couple of brief announcements. First, I want to encourage everyone here to follow us on LinkedIn to get the latest details on our services and events, because we do post there quite frequently. And so to make sure you don't miss out on the next webinar sessions and other events that we might be able to meet with you that we'll be attending. So our next webinar will be in January 2022. And so, yeah, stay tuned there. Or you can also, if you're not on LinkedIn, be sure to subscribe to our email list where you'll get weekly or bi-weekly emails to hear more about our models and services. So with that, I'd like to wish everyone a wonderful holiday season. And if you have any other questions about models and services that we offer, please don't hesitate to reach out to us. We'd love to help you move your project forward and hear about how we can help you. So thank you so much, and everyone have a great day.
Great, thank you.
Thank you, Gina. Thank you, Clark. Thank you, everyone.